Next, we're going to discuss sort of models. Again, jumping gears or shifting gears entirely, maybe not entirely, but at least to a significant extent. One of the things that biochemists and sort of people that are interested in quantitatively studying biology have to do is they have to understand the data that they're collecting in terms of some kind of mathematical behavior. And so this next session, we're going to talk a little bit about how we can understand data using equations, how computers can be used to, extract things. Like, for example, if you want to fit a line to some data, how is the computer doing that? What's the best way to do that? And so this session will assume that you have Excel installed in your computer. We'll be using Excel a lot. Most of you, already have that on there. And, I know MSU students, it's free. I imagine most other universities provide some kind of access to the Office suite. But before we get started with that, let's jump right in with a discussion question. And we're going to start and we're going to say, what is a scientific model? And so once again, we're going to take a couple minutes. We're going to, chat with your neighbor and tell me what you think, in a few minutes, what a scientific model is. So let's discuss. Okay. So what do you guys think? Who has some ideas? I know it's a long day. How about somebody from the Alabama REU? Let's hear how Tide really rolls. Jamie, did you want to suggest something? Okay, yeah. I mean, hey, we're all friends here. Go on back to there. Yeah, hold on a second.
I'm sorry. I said it was includes different types of graphs and tables and pictures in order to showcase information and data from research for others to understand.
Okay, so that's good. I like that. So so basically, in summary, she said, showcasing graphs and tables and highlighting how data sort of works together. And so there's a lot to digest there. Anybody else want to contribute or have suggestions? There's a lot of right answers here. We're going to have different conversations about what model means in this context, but yeah.
I would say it's more of a platform supported by theory and data that other people can follow and add to, as well as reproduce.
Okay, so there are some good things in there. So you added the words theory and reproduce, right? And so one thing is with a model is that you need to have, some way to understand how those data are working together. And so that sort of, builds upon what we had just talked about with understanding sort of tables and, how do those tables and graphs, how are they made? Don't go anywhere, Chithuri. Yep.
We kind of talked about how scientific models can be open to interpretation kind of based on what area you come from. Mm-hmm. So how it affects your research, you may look at it a little differently than someone else.
That's absolutely true too. So again, they can be open to interpretation based on the theory that's involved. And this sort of gets at the fact that all models will have limitations, right? And so, for example, we can imagine the Newtonian physics model for understanding how matter and forces interact. So F equals ma. That's going to be a great model for understanding things like, the speed of a car on a highway. But it's not going to be so great if I'm thinking about photons traveling down some kind of interstellar space, or an electron or a proton in a synchrotron system where I'm approaching the speed of light, right? So models will have limitations based on the assumptions that go into those models, right? So not every, one model may not be applicable under every circumstance. Any other ideas or thoughts? Yep.
So I was thinking, where we were discussing something like a representation of patterns, something about the way you can kind of like she said, reproduce it in different patterns to showcase. So we were talking something along that line.
Well, I think maybe what you're getting at is predictiveness, right? So if it's a pattern, and my theory describes the pattern accurately under the right assumptions, then we should be able to predict the next sequence or the next data point or the next value that, under a different set of experimental conditions. And so this is all really good. This is getting at sort of what a model is. In this context, in the context of this session, for us, in many ways, a model is going to be some kind of equation, right? But it's an equation that is sort of an equation and then some, right? And so if I want to sort of get to models in the context of model fitting or using models to describe my data, right? Data is often measurements that we're making. And so in that sense, a model has to explain some kind of observable quantity, right? So generally, I go into the lab, I make an observation, I come back, I write that in my lab notebook, or even better, I write it while I'm in the lab. And then, I want to use my model to make sense of that particular data that I've measured, right? This could be something as simple as a calibration curve, where maybe I measure a whole bunch of absorbances, and I try to calculate what the extinction coefficient is, for example, using Beer's law, so it's just a straight line. It could be something much, much more complex, where I'm trying to explain, in my lab, we're often trying to explain rotational diffusion of molecular systems. And so we measure a ton of relaxation rates in the NMR, and we have to feed all those numbers into a very complex system to then spit out what we think the rotational diffusion properties are. So a model should be able to take the experimental data and make it more understandable. The other thing that a model needs to be able to do is relate that sort of data that we've observed to what we would generally call parameters, right? So in the case of a calibration curve, your parameter might be the extinction coefficient, right? I can't, we don't have a little, imagine a pH probe where I just sort of shove that into a compound and it reads out the extinction coefficient. There's no way to do that, right? I can't just take a sample probe and stick it into my reaction and tell me what the enthalpy is, right? I have to do experiments and then get some data and then extract the extinction coefficient or the enthalpy of the reaction. And so those parameters are generally the things that we understand, whereas the data, just looking at the data, I'm not going to be able to just look at my NMR data and say, oh, that rotational diffusion coefficient, I understand what that is. No, the rotational diffusion coefficient is a parameter that I extract from the data. And so in this sense, the model relates the experimental observable, the stuff that we measure in the lab, to the parameters or the simplifying numbers that we can make sense out of, either experimental rotational diffusion coefficient, extinction coefficient, enthalpy, equilibrium constants. All of these things are really parameters of scientific models that we don't measure directly, right? Wouldn't it be nice if I could just take my beaker and again, maybe drop in an electrode and say, this is what my equilibrium constant is? Wouldn't that be awesome? really you can do that. I mean, that's really what pH is telling you. It's telling you something about proton activities, but that's like the only time that you can do that, right? So any practical experiment that you want to measure an equilibrium constant, you're going to be using a model to extract that. And the final thing that we should think about with models is that they should be predictive. And so this gets at the pattern idea. If I measure a set of conditions, so for example, if I measure a calibration curve and I say, okay, this much absorption signal corresponds to this much concentration, I should be able to use that model, in this case my linear Beer's law, to say, okay, well now if I have this much absorption signal, what should my concentration be? Right? And so in this sense, we're just using models as a means of understanding the data and then saying, okay, if I change my experimental conditions a little bit, can I still grasp what I want to know? Be it a rotational diffusion coefficient, maybe at a slightly different temperature, or a concentration, maybe at a slightly different absorption coefficient or absorption. So thinking about biochemical models, right? This is sort of what we're here for. We often think about some very simple examples. And so one of these would be protein binding to a small molecule. And this would be the example of your protein binding to zinc, right? So if we have a ligand, which we represent here with L, and we mix it together with the protein, then we model this as a one-to-one binding at equilibrium. And this is a lot of equation and jargon here, but if I want to know the concentration of the bound species, so the PL species, over the total protein, right? So the total protein concentration that I started with, I can derive, and if you're curious, I can tell you how to do this, but I think nobody's curious. I can derive this equation that tells me the observable, which in this case is the concentration of the bound protein, using the initial concentration of the protein in the ligand, and then my parameter here is the equilibrium constant, right? And we'll see examples of this, I think, a little bit later today. Another type of simple biochemical model that gets into some thermodynamics is this idea of the fraction of protein that's folded. And so most of you know that if I start with the protein at room temperature and it's it's not a disordered protein, but it's a functional enzyme, it's going to be active, and eventually if I continue to heat it up, eventually that protein is going to unfold. And so this can tell you the fraction of folded protein, which I represent here F. That's our experimental observable. And it can be related to the enthalpy of unfolding and the entropy of unfolding. So those would be my parameters, and the fraction folded would be my observable. So again, this is getting into a lot of thermodynamics. I don't want to make this into a thermodynamics class, although I love thermo, so you'll have to bear with me. I think the simplest way that we can envision this, though, and a model that maybe we all understand or at least can grasp in this moment, is that if I have radioactive decay, right? And so if I have a nucleus that's undergoing radioactive decay, we all know that there's something called a half-life that tells me how long that radioactive nuclide is going to be around, right? And so this is the radioactive decay of 137M barium. And what you can see is if I stick my Geiger counter up to the barium, I'm going to get a whole bunch of counts, which is, the number of counts is proportional to the number of molecules, or the number of atoms in this case. And eventually, over time, this has a fairly short half-life, but eventually it's going to decay to where I don't really get any counts at all. Or the only counts I'm getting are basically from atmospheric radiation of nuclear particles flying through the atmosphere, which hopefully is a very low number. So how do I go from this, which again, this is the experimental observable, to understanding what the half-life of this barium nucleus is? Which is, that's something that I can understand. That's what I would want to know. That's the parameter. Well, again, the physics would tell us, okay, we have this equation. Again, we're not going to derive any of these equations here, but notice once again, this is relating the observable, the number of counts, is equal to some constant, which is a parameter that we honestly don't care about, plus the background radiation, which we might care about, but we probably don't care about that either. And then the main parameter that we're interested in here is the half-life. Right? And so this is the equation of what you would expect for radioactive decay given a half-life. Now this 0.6931, that's just a conversion to go from half-life to something that's in the log base of E. Again, don't worry about the details here. This is the model, and then this is the data. And again, notice that the model has the potential to describe what the data are doing. It has the potential to predict what the data will be, right? So if I use this model and if I know what the half-life is, I can go out here to like 1500 seconds and I can predict how many counts I should see. Similarly, if I go to one of these spots where I haven't measured, right, I'm getting incremental time points. If I go to this point here in between this point and this point, I should be able to predict how many counts I would see if I were measuring at that time. Right? So my model is telling me something about the data. It's expressed in terms of some parameters, in this case A, the half-life, and B. And it allows me to predict the experimental data under different conditions, in this case, times that I haven't measured. Is everybody with me so far? This is a little bit different way of thinking about models, but once you sort of adopt this kind of framework, this works for all sorts of models. And so we might normally think of a traditional sort of a chemistry ball and stick model, like what you've all played with in organic chemistry. Right? It's not an equation, but it allows you to make predictions about the shapes of molecules. And so the equations are really built in to the shapes of those molecules and the angles that they let you build atoms on. And so, it's explaining the structure, which you can observe indirectly. It's giving you some parameters, oftentimes the angles that you might bend, those hybridized bonds and things like that, those torsion angles, and it's allowing you to predict what the conformation of your molecule is under a certain set of circumstances. Again, many cases, you're often trying to allow the flexibility in those angles to sort of equilibrate and find a situation where your little molecular model isn't strained. Right? And that's telling you kind of the low energy state. Okay, so the question becomes, this is all very well and good, right? I mean, I can sit here and explain, oh yeah, no, we just need to make this, use this to explain this data. The question becomes, how do I do that, right? And this is where computers help us, right? So this is why this isn't a math class. Although we are going to get into some math. I hate to say it. The idea here is that we want to minimize a metric that describes the difference between our observed data and the model using a specific set of parameters that we choose. Right? Now we can go through all the derivation here. And again, I promise I'm not going to kill you on the equations in this section. But I would propose that this value here, which is called chi-squared, is the right metric to tell us the best agreement between our model and the observed data that we might have collected. And so let's go back. We're going to do everything in the context of this barium decay. Right? And so again, this is going to be our model. This is going to be, F of A, the half-life, and B. Right? And so, if I choose a value of A of 1, a half-life of 120 seconds, and B of 50, Well, that's going to be some model. You know, you can imagine I could just draw that on the screen. And that's where we're going to go into Excel and sort of start playing around. So if you go online to the Biochemistry Bootcamp website, you should be able to download a file called decay fitting. And if you open that up, it'll have a picture of our decay window, right? And so this is the data. And I've set it up here, so if we go here, I have a whole bunch of additional stuff that that I'm including here. But mainly the data that we're looking at is in these first two columns. So the first column is time. The second column is the counts in 30 seconds. So that's, again, the y-axis. And then the third column is the error bar, the uncertainty. And a little trick here, if you haven't used Excel extensively, I can go in here to the View panel, and I can go to New Window. And now I can look. In one window, I can be over here diddling with the table of data. And in the other window, I can be over here looking at the graph. And so, you know, I have one workbook that has the data in it, one workbook that has the graph in it. And here, I'm basically looking at the orange line, which represents our model or the equation that we're trying to use, and the black points with error bars, which represents the observed data that we have. And the way I've set this spreadsheet up, and you can set up your own spreadsheets this way, I think this is actually a great way to sort of get familiar or intimate with your data. My group often hears me talk about intimacy with your data. I think you need to sort of know your data better than anybody else, and sort of using Excel, playing with your data. Again, I'm going to use that word play a lot because I feel like that's kind of what we're doing. We're having fun by examining how models and data interact. By using Excel, you can get a really good feeling for how your model and your data work together. And so in this Excel spreadsheet, the way I've represented this is that up here I have A, so that's the initial height or the background. And again, if I go back to my equation, this A corresponds to this A here. I have the half-life, which in this case I've just used the value of 50 seconds. And then I have the background, which I've assumed is 0. Right? And the way I've implemented this Excel spreadsheet is if I change this, so if I make this 110, it's going to alter the shape of this curve in real time. And so if I want to make this, 300, you can see that it basically changes the curve as I change the number. How do I do that? Well, I use some Excel trickery. If you've never seen, who here has seen the $ notation in Excel before? Okay, a good number of people. This is one way where I can type an equation into this cell, for example, and if I use the $signs, like $B, $8, that means if I then go and fill that equation down, it's not going to change B8. It's going to leave that to be B8 throughout all of the equations that I fill down. Whereas if you notice, if I go here, this says A34, and up here it's A15. So Excel is intelligently changing the row number for A15 so that it goes A15, A16, A17, and so forth. And notice that this is kind of the X value, right? A18 corresponds to the time of this particular row. But sometimes you don't want it to do that. Sometimes you want Excel to say, hey, I want to use this value. Don't change it. Don't try to be smart about it. Just keep it to be, in this case, cell B8, which corresponds to my background, B6, which corresponds to my initial height, and B7, which in this case corresponds to my half-life. And I'm assuming that most of you are comfortable enough with Excel that if you look at that equation and you compare it to this equation here, you'd be able to say, ah, yeah, B6 corresponds to A. EXP corresponds to this, lowercase e. Natural log 2 corresponds to this 0.6931. If that's not obvious to you, you can always put it into a calculator and convince yourself that that's what that number is. But anyway, so now I have a situation where I can go in and I can change this. And I can say, okay, well, 310, that looks like it might be a little bit too high. So what if I go and I make it really simple? Let's say if I make it 175. Oh, that looks like it's a little bit better. And now if I make the half-life, let's see. Well, it's clearly longer, right? This is decaying pretty quickly. So it's clearly longer than 50 seconds. And if I want to think about what half-life means, half-life is the time that it takes for half of it to go away. So if I start at 175, half of 175 is going to be about 85, 87. So if I look here, 87, that corresponds to maybe about 180 seconds. So I can go in here and type in 180 and see what happens. Well, that doesn't look too bad. But clearly this isn't the same. If you showed this graph to your advisor or your research advisor, they would say, this doesn't look like a very good fit. How would they know? I don't know. We'll have to think about that one. We'll get back to that. Clearly a computer, when they're doing this type of optimization, is doing it in a little bit smarter way than just saying, yeah, I bet if I make this 200, it might get worse. Oh, that's 20. And maybe if I make it, 150, maybe it'll get a little bit better. But now I have to worry about this background term. What if I make that 20? Well, that's too much. So clearly a computer is doing it in a smarter way than what you and I would do it. But fundamentally, if you can understand what we're doing here, in the sense that we're just changing parameters and looking at the agreement between the model, which is the orange line, and the data, which are the black points, then you've understood everything we're going to talk about today in this class. Right? The rest of it is just algorithms. And again, you don't care about the algorithms. I don't care about the algorithms. What I want is I want a computer program that can figure out how to do this so I don't have to. So, computers clearly have a way to do this. But remember that computers only think in numbers. You and I can look at that, and all those little neurons in the back of our brain, they're saying, yeah, that doesn't look right. I better make this shorter. I better make this bigger. Computers don't have that kind of perception. Everything in a computer has to be reduced to a number. And so, again, the number that I posit is the correct number is chi-squared. That's this. Now, chi-squared, first of all, notice that it's a sum. It's a sum over many terms. And actually, this is the reduced chi-squared, but we'll get to that. Each of those terms, though, is a difference squared. And specifically, let's ignore this 1 over sigma for now. Specifically, it's a difference between the experimental data point, yi, and the data point predicted by your model for a certain set of parameters. Right? So if I go here, back into my Excel spreadsheet, what is this number? Well, this is what we would call a residual. And notice, it's just the difference. It's the difference between the observed value, in this case it's B15, and the calculated value using these model parameters. And so, again, sometimes that's going to be positive. Eventually, if I go back to my graph, in this case, it's almost always going to be positive, or negative, I'm sorry, because the black dots are always less than the orange curve. But if I use a somewhat more realistic value, let's go here to 150. 130. So you can see some of these are now positive, some of these are negative. And one could say simplistically, well, why don't I just, if I'm trying to make this into a number, which is my goal for a computer, why don't I just add up all of these values and just try to make that as small as possible, right? In other words, I'll take those residuals, I'll sum them, and then I'll modify my parameters until I minimize or I find the smallest value of the residuals that I can. Now, why wouldn't that work? So if you look at your slide, we do have the residual here. We have the difference between the observed data and the model. But it's not just the residual, it's squared. So there's a hint. So why can't we just take the residual and sum them up? Nobody's going to think less of you if you're wrong. Certainly not me. In fact, they'll be thankful that they didn't have to answer. So if you have an idea, it's too easy. Very good. I think, well, in some sense it is too easy. Now the question is why? Let's see what we look at. Let's see what happens to our data here. So I'm not sure I'm going to be able to do this easily, but I'll try. Let's try to do this. So I'm going to change this number. So what I'm going to do in this cell is I'm going to hit sum. And I'm going to do the simple trick. I'm going to sum up all of our residuals. Right? Now that's a pretty, it's first of all, it's negative. So we're just going to try to make this as close to 0 as we can. But let's notice something. If I make this, so we're starting out at 282, or minus 282. If I make this 50, yeah, that's worse. So let's go back to 20. Let's make it 25. Oh, that's still worse. But let's make this 100. I can make that 80. Okay, so now what we've done is we've gone from 282 to 94. But is that a better fit? It's not really. So what do you think I've done? What assumption have I made that really isn't applying? How am I gaming the system here? Because I'm gaming the system.
You're assuming that the error in the individual points is the same as the error overall?
I don't think so. Okay, so what you can think of what I'm doing here is I'm taking the difference, right? The residual is the difference between the observed point and the model. Well, that's what our goal is. Our goal is to get the model and the data to be as similar as possible. But if I use this metric where I sum up the differences in the residuals, you can see I can make the absolute value of that sum lower, but my curve hasn't gotten any better, right? I can do it even more. Let's see if we can even make it more extreme. Well, that may or may not be what I'm trying to do. I'm trying to make a point. So let's see if I can get there though. So 180, 150, 30. Okay, so now I've gotten to 60, and maybe this is a little bit more obvious. Again, what I'm doing is I'm summing the difference between the model and the data. So how am I gaming the system? I ask again. Well, that's getting closer, right? So notice how all these points are above the line and all these points are below the line. Yeah?
You can have substantial error in your graph and your trend line. It doesn't matter as long as the overall is 0.
Yeah, exactly. So I need to make sure if I use the residuals as my metric that the difference between all of these points, which is going to be positive, is matched by the difference between all of these points, which is going to be negative. And that's the problem. Because as long, I could imagine drawing a straight line through here, and I can get the sum of residuals to be 0, but all I've really done is made sure that the area under this curve is equal and opposite to the area under this curve. So that's the problem. That's why we can't use just the residuals. What happens when we use the residuals squared? So if I square a negative number, what happens to it? It becomes positive. So once I square a negative number, there's no more way to game the system, right? If there's a difference between the experimental data and the model, and it's a large difference, if it's big, it's going to be a large contribution. If it's small, it's going to be a small contribution. If it's small and positive, or if it's small and negative, it's going to be a small contribution. If it's big and positive, or it's big and negative, it's going to be a big contribution. And so if I instead look at those differences now squared, which is really all I'm doing here, I'm taking the residual and I'm squaring it. Notice this is E15 squared. Now I can't game the system anymore. And if I make this small, right, let's try to make it a little bit smaller. So let's do 26.7. Right? So this is going down, but the chi-squareds are getting all getting bigger, or the residuals squared are all getting bigger. And notice there's no negative values in here. So if I sum all these up, I'm going to get something that's always going to be positive. Right? So now let's try to do this a little bit more sensibly. Madison, what values did you get? Okay, so now if we look at these chi-squared values, they're actually all pretty small. Notice that the residuals, some are negative, some are positive, but the chi-squared contribution is always going to be small. And here's the sum down here at the bottom. I just summed them all up and divided by the degrees of freedom. Again, we're not going to worry about degrees of freedom here. I want you to understand where this is coming from. But so now, and then that value is now here, it's 2. Right? Now, but I can still make it better, I bet. And maybe I should just do this as a competition. And maybe that's the solution, right? Go to something like Google's Mechanical Turk and say, hey, find me the best values for this fit. No, we don't want to do that. Computers can do it better. But notice if I make this 5, my chi-squared sum has gone down to 1.3, so it's a little bit better still. If I make this 10, now it's gone up again. So now let's leave it at 5. And let's see about, what happens if we reduce the first point again? Well, now we go to 170. Ooh, that's getting better. That actually almost looks like a reasonable fit. And so if I think about what this chi-squared term means, we're going to try to minimize this. We minimize the square of the residuals, because we can't game the system that way. We also divide by the uncertainty squared, right? And the idea here is that if I have a large uncertainty in a data point, I expect that my model and my data point are going to differ probably on average by my uncertainty, right? If it's a real accurate value of the uncertainty, if I measure an inch plus or minus 0.1 inches, and somebody else is measuring an inch, I expect them to probably be within 0.1 inches either way, right? And so if I divide by this, it's just a constant. It's going to be it's just going to scale my chi-squared down. But in this sense, it means that I expect my numerator to be approximately equal to my denominator, right? So on average, the difference between my experimental observable and my data point should be on the same order of magnitude as the uncertainty. And if I look at this term for the first data point, I should get something like 3 squared divided by 3 squared if my uncertainty is 3. My second point might be off by 2.5, but it's going to be off by about the same value for every single data point. And so if I sum up over those, what would I expect chi-squared to be if my data points are on average the uncertainty or different by the uncertainty of my measurement? The wheels are turning. So what's this value going to be for the first point? Approximately. What would I expect it to be if the numerator and denominator are equal? What should I expect that to be? 1. What should it be for my second point? 1. What should it be for my third point? 1. It's going to be about 1 for every point. Provided my uncertainty is reasonable, every point should probably be, on average, different from the experimental data by about 1 standard deviation or 1 level of sigma. And so for the first point, I get 1. For the second point, I get 1. For the third point, I get 1. And so the sum of this should be approximately equal to the number of data points, right? Now, there's some exceptions there because we're fitting a model, right? So we don't just have a pure system where I'm just throwing out data points and expecting them to agree. I have parameters that I'm using and I'm trying to optimize. And so those sort of go into the sum, and that's why we divide by what's called the number of degrees of freedom. There's nothing magic about that. It's just the number of experimental observations you've made minus the number of parameters that you're fitting. So for this model, how many parameters do we have? 3. We have the baseline, we have the half-life, and we have the initial intensity, the A. I can go back here into Excel, and I can count. And so I have 40 rows, so I have 40 experimental observations. And so then what's my number of degrees of freedom? You can read it right off. 37. So 40 experimental observations minus 3 parameters. And if I take this chi-squared value and I optimize the parameters to be as close as they can get, provided my uncertainties are realistic, this should be about equal to the number of data points, about 40. This is going to be equal to the number of degrees of freedom, so in this case 37. And if my system is well optimized, my chi-squared should be about equal to 1, or my reduced chi-squared. So the reduced chi-squared means that I'm dividing by the number of degrees of freedom. Again, if I'm just minimizing a number, I can minimize this thing and I'll get a perfectly reasonable answer, right? But if I'm trying to quantify that statistically, that's why people often reduce it by the number of observations. And so now I can go in here. Is this a good chi-squared? Yeah, I mean, it's about equal to 1. So, and if I look at my data, yeah, it's pretty good too. Now here's a trick that's going to blow your mind. Maybe, maybe not. How many of you have ever used Excel to optimize data before, or optimize functions before? 2, maybe 3. I've set this up very specifically so that I could change these values and I could look at this value and try to minimize it. But that's a computer problem. That's not a problem that you and I are particularly good at, but a computer is actually really, really good at that kind of thing, because it basically just means, scanning through these values, and once you have a metric, you can tell the computer, hey, find the minimum value of that. And so if I go here into Excel, Options, and I go into the add-ins, and I'm doing this because your Excel might not have this enabled, and this one doesn't. So you'll see that this one doesn't have it, but I click on this Solver add-in, I hit OK. Oh, please work. Well, this will be good. Hopefully, if you guys are running in this window, it'll pace us a little bit. Okay, good. So now if I go here into the Data tab, you'll notice I have a new feature called Solver. And I'm going to click on Solver, and it's going to bring up this window here where I can set an objective, and I can set by changing variable cells. Yours probably won't be set with this, but maybe it will be. In this case, my objective is going to be the reduced chi-squared. Right? That's the number that you want to minimize. And notice I've set that up by how I made my Excel spreadsheet to begin with. Right? I have my observed values, I have my calculated model, I have my parameters up here. Right? So some care went into making this a spreadsheet so that it would work like this. But it's nothing that you guys can't do in an afternoon. And one of the reasons we give you this spreadsheet is so that you can sort of see it and use it as a model. OK, now I want to minimize that, so I'm going to make sure that the minimum button is clicked. And I want to change the variable cells that correspond to my parameters. So I'll hit OK. And now I'm ready to go. I don't really have to change anything else here. All of these I expect to be positive, so I don't have to do any weird checkboxes or anything. You guys can read this, but generally you can leave this section blank. And I'm going to hit Solve. And notice that it's changed the parameters. So these are no longer the values that Madison gave me plus my tweaks. The chi-squared has now gotten even better. So it's 0.4. So the lower your reduced chi-squared is, generally the better the agreement is between your model and your data. And if I look now at my graph, that actually looks pretty respectable. And this is something that you could then say, OK, my best estimate for the half-life of barium-137M is 144.6 seconds. And I can back that up with the chi-squared. And I can talk to my research advisor and say, OK, well now, how can I estimate the uncertainties? There's a lot of different rules on that. For my group, one of the very common ways that we do this is we would actually repeat this experiment 3 times and calculate the standard error of the mean. So I would measure the half-life 3 different times and then measure the standard error of the mean, and that's how I would report my uncertainty. That's usually better than trying to estimate the uncertainty based on the fit. One, you never know if, maybe I didn't measure barium-137, maybe I measured barium-138, right? And so if there was something pathologically wrong with your experiment, and you only use one data set to express the half-life in this case, or the parameter, whatever you're measuring, you might be, there might be something very wrong, and if you repeat the experiment, you might get a very different value. So that's why we usually emphasize, if you can repeat the data and measure it again. Okay, questions on what we've covered so far? It's a little mathy, but hopefully applied enough that it's holding your interest. There's no way we're going to get through this whole talk, so don't worry about that. So what are we doing when we minimize chi-squared? I mean, and so this is again getting a little bit into the algorithm side of things. But really what you could do is you could imagine that I'm varying a parameter. So in this case, the parameter is k. But you could imagine this being the half-life or the A or the B value. You're taking that value and you're basically making a graph. You're plotting chi-squared versus that parameter. And you're trying to find the position where that parameter is minimized. And in the case of our example with the barium decay, we would have 3 dimensions. 3 dimensions are hard to visualize, so I've drawn this as 2 dimensions. But you could imagine that if I had slope of a line and intercept of a line, I would basically be trying to optimize my curve so that I can locate this position of chi-squared where my slope is best and my intercept is best. Or in the case of the barium, where my half-life, A, and B values are all the best. And again, fundamentally, this is what a computer is doing when you're optimizing a model. And so this would be our best value. This should agree very well with what we just came up with. And so rather than discussing, I think we'll just push on. Now we get to the question of, is this fit any good? Right? And so what's the ways that we can look at? And these are ways that we've already talked about. The first way is look at the plot. Right? When we do if you're in a situation where you can directly compare a curve or a predicted curve with your data, do that. Make that plot in Excel and look at it and see if it agrees. If you don't use Excel, use whatever program you're using. Just look at the curve and say, yeah, no, that seems like it goes through all the points. And if there are error bars, I might be slightly outside of the points every once in a while. But if there are error bars, make sure that the error is about the same order of magnitude as the difference between the curve and the fit. Every once in a while, you're going to see something that shows up. But again, if you roll the dice enough, you're going to see every once in a while the data aren't going to agree very well either. And so that's where the statistics, that's where randomness come in. The other thing that we can use when assessing a fit is asking ourselves, what's the reduced chi-squared? If the reduced chi-squared is less than 1, and you're confident that these error bars are good, then that should indicate that your fit or your agreement between your model and your data is acceptable. So that's good. And then the final way that you can use to assess the fit is by plotting those residuals themselves. And so if you go over into my original spreadsheet, I calculate the residual and I use it in my calculation of the chi-squared. But I also have a graph here where I just plot those. And if I look at the residuals, yeah, they are roughly scattered around 0. When they differ by 0, it's usually not by more than an error bar or 2. And if I look, there's no systematic trends, right? This side isn't systematically higher than this side. Or there's no big curve in my residuals. And we'll see some examples of bad residuals here in a couple of slides. Right? So this is the bad residual. Notice how, yeah, things agree here at the end. That's pretty reasonable. But all of the points here in the first 400 seconds are less than 1 or less than 0. So that means that my model over-predicts the data. And so by plotting the residuals, I might not be able to tell that if I look at the graph directly. But if I plot the residuals, since I've sort of reduced it to a very small scale, I can say, oh yeah, those residuals don't look so good. Here's a really bad one where the residuals start out close to 0, but then they make this huge jump, and now they have this other weird thing. So again, if my residuals look like this, they're not randomly scattered around the y-axis. And clearly there's some systematic problem in my model. Okay. So what can we do in our remaining time? We have about 10 minutes. The first thing I'm going to do is, now that we've talked about model fitting, and notice I'm always saying model fitting. One of the common misconceptions or misstatements that I hear is people will say, oh, I'm going to fit this data to a model, right? But generally, you're not fitting your data. If you are, you're probably not treating it correctly. So you're fitting the model to the data. That's the appropriate wordage or verbiage. Now that we've talked about model fitting, you guys can go into this Gnuplot tutorial. And in this case, hopefully you all have, I've disabled those lockouts on the Linux server. And so if you go here, you can either go on to Linux and open Gnuplot that way, or this is one of those examples where if I just you can install a Windows version of Gnuplot, and it'll bring up a fairly nice window that you can work with. But now that we've talked about model fitting, you can go into this Gnuplot for chemists, and you can understand how these terms are being used. And the really nice advantage of Gnuplot, I talked about ways to estimate the uncertainty of the parameters using the error bars that you've already collected, right? I'm not going to get into how that's done because the math gets a little hairy. But Gnuplot has the way to do this digitally. And so in this case, in the Gnuplot tutorial, you'll be fitting a line through some data points. You'll generate this plot where you can look at the residuals. But then when you do Gnuplot, you'll be able to look at this, and it gives you a value. So it says, my best estimate for the slope is 4.5 plus or minus 0.02. So it gives you some uncertainties of the parameters. And you could do this with the exponential decay, and it would tell you approximately what the uncertainty in your half-life is, the uncertainty in your baseline is, et cetera. So Gnuplot has a convenient way of doing this. The other thing with Gnuplot, and we'll get to see this a little bit in the Python section as well, is how do you work with Gnuplot, right? Lots of people, so Gnuplot has this nice terminal, and I can type things like I typed earlier. I can type plot sine of x, and it'll generate this nice plot of sine of x. But generally speaking, you should be able to have, you shouldn't have to type every single thing, because this gets very cumbersome. Most Linux terminals, if I press the up arrow, you'll see that it brings up the previous statement. But that's still too slow, and most people are pretty lazy, including myself. And so, for example, if I want to do something in Gnuplot, I can go up here into the directory, and I'm going to browse to my desktop, and that means every file it looks for will be taken from my desktop. And if you go to the website, you should be able to download, for example, the linear data. And so from within here, I can type plot linear.txt. The W stands for width, Y error bars. Oops. I thought that was how it worked. Well, let's try getting rid of that first. Okay, so that works. Good old Adobe. Anyway, so from within here, though, again, I don't want to have to do all this plotting typing at the command line. The easiest way to do this kind of thing is to open up a text document. We'll call it myplot.txt. And if I type that same statement in here, so let's do plot linear.txt. And in this case, I'm going to give it a comma, and I'll say, let's do 4 times x plus 3. So now I'm plotting two things. I'm plotting the context of linear.txt and the function 4x plus 3. And I'm going to type load myplot.txt. And so you can see that now I'm in a situation where I can type into this window. So this screen's a little small, but we'll make do. And now let's suppose I want to make a change, right? So now I don't have to go back into this window. I can go, okay, let's make this 4.5x times 3. And I'll save that. I'm going to add another statement at the top, which is basically resets all the graph stuff. And so now if I go in and type and press up, my word, you can see that I can basically make my changes here and then go back into the window, reload the file, and look at the result. And for lots of programs, like we'll see this for Gnuplot, this is by far the easiest way to work with this program. We'll see this for Python. We'll do the same type of thing in Python where we'll have a text file open and we'll use an interpreter to reload that text file over and over again. And this way, if you want to make some changes or you want to sort of play around with how Gnuplot works, you don't have to keep typing everything over and over again. You can go into the text file and just make the changes that you want. And so, again, now that we've covered what model fitting is, we've covered sort of how to represent data, we've talked about uncertainties, we've talked about residuals, hopefully you can go through this and it'll make more sense than it would have if I just said, go try this. But if you have an assignment here tonight, this would be what I would recommend, is try working through this. The fact that you don't have to log into Linux means that you should be able to do it without too much trouble. On the other hand, if I opened up my Xterm, and so for those of you using Windows who log into WinSCP, this is, it's a very convenient way to copy files back and forth, right? So in this case, I can copy my linear.txt. I can even copy my plot. And so now it appears on my Unix workspace. Let's copy linear.txt as well. And I can do the same thing here, right? No, I don't want that open. So I'm going to open up my editor, which in this case is going to be Xemacs. I have to spell it right. I'm going to terminate it with an ampersand because I want my command line interface back. And so here I can open my myplot.txt. Notice it's the same file. It's reset and then plot linear.txt. Xemacs has its own sort of unique features, so I recommend if you're new to it, use the window to save and whatnot. But I can start Gnuplot here. And from the current directory, I can now type load myplot.txt. In this case, I can use tab completion. So you notice if I just type myplot and I hit tab, it automatically completes it for me. And now it's the exact same window, but it's running on my Linux system in the building next door. Right? And so if you don't want to install Gnuplot, you're welcome to use the Gnuplot on Linux, and it would be one way to practice how you might get access to things. And then that Gnuplot tutorial will walk you through doing basic plotting, doing model optimization, plotting residuals, getting parameters out. Instead of using the exponential decay, it uses a linear fit, which is a little bit easier to manage, but it's the same principle. Okay, questions on that? So we skipped a few slides here in this presentation, and that's fine. There's no reason we have to go through all this. There's basically some additional stuff on how you think about building models from scratch. And so you can imagine that this really gets into the physical chemistry side of things. And so I would call myself a physical chemist because I try to work from first principles to develop those equations that understand how biomolecules behave. And so there's lots of thermodynamics that goes on in my work. Obviously, all of the NMR has to involve equations somehow. And so we're using a lot of that material. But there's definitely people who aren't physical chemists that still need to be able to build models, in which case, what if I have, protein that can bind zinc and manganese, or maybe, it can bind one DNA or another DNA. How would you understand how that works, right? And so you would have to sort of go through those equations and figure out what equation would best describe the values that you can observe. And often that's really hard. Right? Building models, particularly if you're an undergraduate or even a very new graduate student, nobody expects that to be easy. And that's something that you would generally get to develop as you become a graduate, more mature graduate student, and as you work towards your postdoc, you develop those skills. At this stage, many of you will be working with your research advisors to come up with that information. But generally, if you're trying to build a new model, you're going to be doing the exact same kind of things that we were maybe talking about. You're going to come up with a hypothesis about how I expect my molecule to behave under different conditions. You're going to try to use first principles, either thermodynamics, kinetics, to develop a set of equations that can predict something that you can look at experimentally. And then you try to do that experiment and compare the fit and see if the parameters that you get make sense. Right? And as the final sort of example, we worked out, or in the slides, if you want to look through them, we're doing a very practical case where I have this molecule called TMPYP4, and it's binding a series of, a G quadruplex, so a series of nucleic acids that form this very specific structure. And you could imagine that maybe I don't know how many TMPYP4 molecules bind one of these structures. You could imagine maybe it fits in between, maybe one binds up here at the top and one at the bottom. But that would be an example of a parameter that has a very fundamental understanding. How many molecules of TMPYP4 bind per quadruplex? And it'd be one that I could really only access via model fitting and optimizing the parameters and seeing which is best. And so I have this Excel spreadsheet online. You can sort of see how I worked at it. But here's some example data. And here's where I've done the best fit model with 3 sites, 4 sites, and 5 sites. And you can see, if you go into the Excel spreadsheet, which of those has the best chi-squared value. And that would be ultimately the one that you would propose as the model for, I think that there's 4 sites. I think that's the best here. So 4 fits better than 3 or 5. But you can appreciate that there's a lot of complexity going on here. It doesn't take a ton of computer skills, but you need to have a little bit of computer skills so that you can sort of frame the question in an appropriate way. And so, yeah, our summary here is basically we've talked about models. We've talked about how to fit data, or fit models to data. See, I did it myself. And We talked about how you can use Gnuplot or, even better, Excel, something that you all have pretty good familiarity with, to really do some advanced analysis of your data. So don't be afraid to play with Excel. Excel is extremely powerful, and you don't need a multi-$1,000 software package like Origin to do most of the things that you can really just do in Excel. Origin's useful for some things, but you can make a lot of headway using a program like Excel. Okay, it's been a long day. Questions? What are you guys thinking? I mean, is there stuff that you're sort of mulling over, or this would be a great time to hey, maybe tomorrow we can talk a little bit about this, or we have this discussion half an hour built into the end of the day. Most of the time, people just book, and that's fine. I'm not offended at all. On the other hand, if there's questions or I don't understand why we care about computers, or I don't understand why where you're getting your error bars for your data from. Those are the types of things that we can sort of talk now if you guys want to have a discussion, and it can be more of a round table too. Yeah.
So the main test we used to fit models was the chi-squared, and does that tend to be the most common test in scientific fields, or is there other types of tests to fit models?
So chi-squared is by far the one that I'm most familiar with. Are there other people that have used other types of tests to compare? What? I couldn't quite hear that. Well, I'm not sure that microphone's going to help. So just speak up.
So p-value and all that?
So chi-squared, so we didn't talk about this, but I can always plug my grad class. There's actually a really great book by a guy called Taylor, and I think it's called error analysis. There's a picture of a train that crashed on the front of it. And I use this for my textbook, or one of my textbooks for my graduate class. All of these chi-squared values are associated with what's called a chi-squared distribution. And so we've used a very hand-waving description to say that the reduced chi-squared should be roughly 1. There is a much more rigorous mathematical way to talk about chi-squared that allows you to associate a p-value, in which case you could say, what's the probability of this data being in agreement with this model and having this chi-squared value? And if that p-value is above a certain threshold, you can say, well, then this model likely doesn't fit this data. But that's, you can either look those tables up in the back of a book, and Taylor is great for this because it has a big table, or most of these computer programs will actually spit that out. And that's one thing that I'll concede Origin does give you that you don't necessarily get from Gnuplot. Okay. Well, thanks for being good sports. Get rest. Go do something. I mean, Starkville's not a crazy, those of you who are guests, there's not a ton of stuff to do in Starkville. But get outside, enjoy some company of other people. We've been in this dark room all day pretty much. So go outside, get some vitamin D, and come back hopefully ready and energized to have another day. And we'll see you tomorrow.